Seems there's a lot of due dates, a lot of confusion the past couple weeks. But I'll just remind you, if you didn't do something, just turn it in late. The sooner you turn it in, the better. Some people even messed up and wrote a report on the wrong lab. So if you're one of those people, and I forgot to mention this with one of them because I've already had to give them a 0 technically on mine for that. If you wrote the lab report on the wrong lab, just write it on the correct lab and consider the wrong lab that you wrote a lab report on extra credit. And I'll get back to you if you're one of the people, because I have mentioned earlier in the semester that if you write lab reports on the other labs, I consider that extra credit. So bad news is you did the wrong lab report or a lab report on the incorrect lab. The good news is you will get some extra credit for it, but make sure you get the correct lab report done. If you have any questions, remember, if you go to the course announcements, since it was such an important lab, there's a whole section under it, under course announcements. It says something like photosynthesis respiration lab. And if you go there, it gives you the video of when we met and I taught you how to do a lab report. It shows you where the due dates are, links to the form, all the stuff you need. But even then, if you can't find what you need, just let me know and I'll help you. And again, a lot of you are missing stuff off the set. It's been a strange couple of weeks. I've never, for you guys, this is the most missed assignments I've seen. So hopefully this is just a weird situation and it's nothing permanent. But again, I'm here to help you. Let me know. Now let's do some attendance. And again, as I'll remind you, because it's this whole identity theft situation and some other things going on, I'm very behind on emails and grading. So if you need me in a hurry, meet me online during office hours or call my office so I can take care of you immediately. Because if you send me an email, I gotta get through all those other emails before I get to your email. Just keep that in mind. All right, let's see, 10:23. Happy Avogadro Day. You can look that up for independent work if you want. That's a one question, one of those, just one question, send me the answer to it. When is Avogadro Day? And in the process of looking up when Avogadro Day is, you'll figure out what Avogadro Day is. It's a chemistry thing, but you know, we can't have biology without chemistry. Anyway, is Aki here? Nope, Aki's not here. Boggs is here. WBSU here. Bonner is not here. Clay is not here. Dawson is here. In WBSU here? Nope, I just checking. Ellis is here. Paula is not here. Dean is not here. Harrison is here, WBSU here. Henson is here, NWBSU here. James is not here. Kesterson is here, NWBSU here. King is not here. Lewis is not here. Linda Q is not here. Morrison is here. No?
No, WBSU.
Oh, all right, I got you. You read my mind. I thought you meant like you're not here, like you're physically here but not mentally here. It is early. Anyway, Grace is not here. Ward is not here. Wilcox is here. Wilder is not here. Parks is here. Really quickly, let me take a gander at who's online. Grace, Paula, James, Ward, and Bonner. So really quickly, let's take a look here. Bonner's online. Don't forget if you're online to send us words when that happens. Paula's online. James is online. Grace, I believe I said, is online. Yeah, I think I got everybody. Also, another thing about this last week lab, apparently it was harder than we thought, right? So even ignoring the fact that a lot of people just didn't turn it in at time, a lot of people got a really bad grade, but don't worry, it's not permanent. In this lab, more than any of them, it seems like you need my help. So when you're ready to redo your lab, if you want to return it in, let me know. We can meet online. Seems like most of the problems were with the Punnett squares And also that second table, you need to indicate, which of the two genotypes you needed to use, or that you used to come up with your frequencies. But again, it's all temporary. If you didn't get a good grade, don't stress out about it. Just meet me online and we'll fix it. That being said, let us jump back into the lecture. The first word for attendance will be the one I'm pointing at right now with the little on the screen. It's the first word for attendance. Starts with a P, ends with an E. Oh yeah, I know a good reminder. I haven't reminded you guys of this in a while. So remember, on Veterans Day, I'm giving myself a day off, meaning I'm not going to come into campus. So Veterans Day, Monday, I think it's definitely the 11th. Pretty sure that's the Monday. We will meet online only for lecture. We won't be meeting here. What was the other one? Lab. Again, this week's lab is completely virtual. So I'll be in here if you need me. I'll be online if you need me. You can come in if you want, or you can work on it on your own. And just as a reminder, the first half is just, you're going to go through a virtual DNA extraction, and you're going to answer some questions while you do that. Make sure those questions come from the screen on the experiment. That's where the answers come from. At the end, you're going to take a picture of your screen to prove that you went through the experiment. And then the second part of it is watching a video while you answer some questions. So pretty simple, probably one of the easier labs that we do. But again, nonetheless, I will be here if you need me. For that matter, like I was just saying, if you really messed up the last lab and need some help with the last lab, you can use today's lab time to get help if you want. Anyway, let's go back into this. So for a large majority of this chapter, we spend a lot of time with that first bullet point. We talked a lot about regular old plain genetics, meaning we talked about complete dominance, where we were looking at one gene that had only two different alleles, and one of the alleles was either completely dominant or was recessive. Then we got to this section. And in this section, I told you that despite all the things we learned in that first section, those are more like the exception to the rule than the rule. Because more often than not, we actually have variations on Mendelsohn's. More often than not, it's not as simple as the things that we taught you in lab and in this first section, right? It's a little bit more complicated. For example, the first thing we talked about was incomplete dominance. And to remind you, that's when if you have a heterozygous individual who has a dominant allele and a recessive allele, they have a blending of the two different phenotypes. So for example, red and pink flowers, a red flower and a white flower would make a pink flower, because that's a blending of the two. Then we talked about incomplete dominance. And I really do need to update this PowerPoint, because yes, that was the example that I gave, but it was the blood groups. But the more important thing you needed to understand was incomplete dominance. Codominance. Codominance is the thing with blood groups. And that's when both phenotypes show up. So again, using the same example I just used for incomplete dominance. If you had a red flower and a white flower, it would result in a red flower with white spots, as opposed to a pink flower. Then we talked about pleiotropy. The example I gave for that was sickle cell syndrome, or sickle cell. And that's a situation when you have one gene, instead of one gene controlling one character, it controls a lot of different characters. Right? So in that example for sickle cell, that one gene causes one protein to be misfolded. But therefore, the whole cell is misfolded, and then therefore there's this whole list of different symptoms that you have with that disease. Finally, that brings us to this. Polygenic inheritance is basically the opposite of what I just talked about, which is pleiotropy. Polygenic inheritance, it's actually kind of in the name. And I just told you it's the opposite, basically the opposite of pleiotropy. So pleiotropy is one gene that controls a lot of different characters. What do you think polygenic inheritance is? And it's kind of in the name. Polygenic. Multiple genes, yeah. Multiple genes for one character, as opposed to one gene and multiple characters. Very good. Polygenic inheritance is the additive effects of two or more genes on a single phenotype. And again, you know how I am with, hopefully at this point you know how I am with definitions. Like, yes, that's a correct definition, but there's the way I just described it. So you don't need to ever memorize definitions verbatim. Just make sure you understand the concepts. So again, what did I say for polygenic inheritance? I said it was more than one gene on one character, right? So same thing, just say it in a different way, which is another good reminder for easy independent work. Verify that. Just look it up. I looked it up, and according to this source, this is the definition of polygenic inheritance. Boom, 1 point, right? If you keep doing this as I'm recommending them, I mean, it's going to be a lot of work on my end, but eventually you're going to start getting those points because that's easy. And again, if you want to, you can write a paper on it. But if you're just looking for that quick point, find another source that says what I'm saying or disputes what I'm saying. Anyway, like I already said earlier, polygenic inheritance is basically the opposite of pleiotropy because, again, pleiotropy is when you have one gene that controls several characters. Here's a big thing for the exam. This is a big, important bullet point. If you were to graph all the different phenotypes, then you would get a bell curve. And I believe on the question on the exam, that's how, I think that's, there's a lot of ways to explain polygenic inheritance, obviously, because there's a whole slide here, but I think that's the way that it's focused on the exam. And I'll give you an example really quickly. And you did do this in lab, and this is one of the reasons you did this in lab. Let's say you're talking about, I don't know, chin dimples. And there wasn't that much of a difference, right? Chin dimples, if you were to graph it, there's people who have it, and there's people who don't. Now, of course, the bars might be the other way, but you see that's pretty simple, right? 2 bars, because you either have it or you don't. You guys with me so far? If you were to graph every human on Earth and put, like, whether they have chin dimples or not, it would only have 2 bars. Right? You gotta hope you're with me on that. This is only you either have it or you don't. With polygenic inheritance, it would be a bell curve. And a good example of that will be height. And this is even an oversimplified version. But imagine, if you were to graph everybody on Earth, it's not like everybody is either short, like, I don't know, everybody on Earth is either 5 feet or 6 feet, right? That's not the case. We have everything in between. So even this graph and this Punnett square, that is a Punnett square, even this is an oversimplification, as you can imagine, right? So for humans, there's tall and short and everywhere in between. And yes, usually a lot more people are in the middle than towards the edges there. So once again, you should expect a bell curve if you were looking at polygenic inheritance because you're looking at a lot of different phenotypes. And again, this might look really complicated and you'll never see this again on the exam. But think about this once again. You've seen a monohybrid cross, and that's what you guys did in lab, right? Where you did a Punnett square. You saw the monohybrid cross. Then you saw the large one, the dihybrid cross, where you're looking at 2 alleles, right? And then this one, as big as it is, this is only a trihybrid cross. This is only 3 alleles. And once again, this is an oversimplification because height is controlled by much more than 3 genes. And also it's controlled by stuff other than just genetics as well. So this example is an oversimplification. Do not memorize the fact that height was the example because I'm not going to use this example, I don't think, in the exam. And again, this is an oversimplification. But yeah, this is the same Punnett square you guys have already done, just much larger. Anyway, any questions about polygenic inheritance? All right, so like I just said, once again, this thing about height is an oversimplification for 2 reasons. 1, there are more than 3 genes involved, and this only shows 3 genes. And 2, there's much more than just genetics involved. So what else do you think could affect someone's height other than their genetics? Like let's say you took 2 twins, and you can look this up for independent work too. Some version of this has happened. Science used to be a lot less moral and ethical. So I think there was some study where they separated twins at birth and then looked at how they were different much later in life. I don't remember in which ways, but anyway, if you were to separate 2 twins, 2 identical twins who have the same exact genes and give them 2 completely different lifestyles, they might be different heights. And what are some of the things that would contribute to their height?
Nutrition.
Good, nutrition. I mean, amongst other things. So keep that in mind when I teach you this new one, which is called epigenetics. So this is the last of the variation of Mendel's laws that we're going to talk about, epigenetics. So many characters, height, for example, result from a combination of heredity and the environment. So when he said nutrition, that falls under the environment, right? So what are they being fed? Good nutrition versus bad nutrition in that example. So that's basically what epigenetics is, when we're looking at stuff that's not just genetics, but it's also the environment. There's a lot of things where this is up for debate. And a lot of times people argue about it. You know, like, were they born that way? Was that genetic? Or was it because of the way they were raised? Or was it a mixture of both? And I'm not even going to get into that, but I guess if you want to, you could look into some of that for independent work if you want. You could write papers about that if you're interested in it. Then again, I won't even say which ones, but there's a lot of debates out there where people say, oh, they're born that way, they can't help it, or oh, they were raised that way, it's their parents' fault. Or usually the scientists chime in and say it's probably a little bit of both. But it depends on what you're looking at. And then the old textbook, and I think even your textbook does give a great example. When you spend time with identical twins, like if you spend a lot of time with them, not if you just met them, if you spend enough time with them, you can probably tell the two apart because they are a little bit different. So despite having the same exact genes, all their genes have not been turned on and off the same ways. They've not been expressed the same ways. Therefore, they are slightly different. How many of you have know some twins where you can tell them apart, or some identical twins? Because I remember there was a question. Boggs has a little bit. I remember there was a question earlier in the semester where I asked you guys if you knew twins. Maybe that was an attendance word, but anyway. So I guess that's not a good example for you guys, since only one of you has that, but kind of. So in general, it's only the genetic influences that are inherited, right? So generally speaking, when we're talking about nature versus nurture, right? Whether we're talking about genetics versus environment, generally speaking, it's only the genes that are inherited, right? So we'll go back to that picture that I first started this chapter with, and I showed you that woman who had tattoos, and she had like, I assume fake red hair because it didn't look like a natural color, it looked really fake. Anyway, yeah, she changed her hair color. Yeah, she gave herself tattoos, but that's not a genetic influence, right? So if she were to have a child, that child's not going to pop out of the womb with tattoos, right? So again, in general, it's only the genetic influences that are inherited. And usually the effects of the environment are not passed to the next generation, whether that be good or bad, right? So once again, I don't know, if you had a father or a mother, whatever, if you had a parent who just really was into fitness, right? They work out all the time, they're really muscular, they're really in shape. When their baby's born, that has nothing to do with it, right? Now granted, if they're really into it and that baby's gonna live with them, they'll probably grow up with that lifestyle, and they'll also be healthy later, maybe. But that wasn't passed on genetically, right? That was passed on culturally, environmentally. However, in recent years, I guess it's not really recent anymore, I should update this too, because this has been a decade ago when I first read about this. But anyway, biologists have begun to recognize the importance of epigenetic inheritance, which is the transmissions of traits by mechanisms not directly involving the DNA sequence. Let me try to break that down for you to help you understand what I'm saying here. Again, only the genetics are the things that gets passed down, right? The effects of the environment, generally speaking, do not get passed down to the next generation. What we're saying here with epigenetics is sometimes your environment can change your genetics. The caveat here is, though, it doesn't do so by changing your DNA sequence. Now, I'll give you some examples on the next slide. So I want to make sure you understand that first. Now, so I'll back up a step two. Sometimes your environment can change your DNA sequence, right? So, you know, if you get exposed to a lot of radiation, if you get exposed to a carcinogen, that might mutate your DNA. And if it's mutated in the areas where you make your gametes, your sperm and your egg, that mutation will get passed down to the new generation. But what we're talking about here in epigenetics is it changes your genetics, but doesn't change your DNA sequence. And again, I'll give you another slide, example on the next slide, but here's my best way of thinking of it. Think about your chromosomes as an book of instructions. Because as you know, each gene is a set of directions on how to build a protein, right? So think of all your chromosomes, all 23 pairs of your chromosomes. Each one of them is a book that has instructions on how to build proteins. So what will happen through the environment is sometimes that book is messed up. Not the words in the book, but sometimes maybe the pages get stuck together. So therefore, it's a little bit harder to get to that gene to produce that protein. Or have you ever taken a book and just kind of laid it down open like this so you don't lose the page? and it stays there too long, and then it kind of always wants to flop over to that page. You kind of think of the same thing with your chromosomes, right? So again, sometimes it's hard to get to some of the parts on your chromosomes. Sometimes it's really too easy to get to parts on your chromosomes. So to me, I think that's the best way to think about it. You're not changing the words in the book, but you're changing how easy or hard it is to get to some of those pages. So anyway, let's talk about some examples. So yeah, the example, this is actually, I kind of had this backwards. What I said was an example. This is more specifically what actually happens. What happens is that chromosomes can be chemically modified by adding or removing chemical groups on the DNA and or the protein components. And this is a good time to remind you that I told you earlier in the semester that chromosomes are made up of about half DNA and half protein. I told you that the DNA wraps around that protein, and those little things are called nucleosomes. And I told you later in the semester this is going to be important. So this is when it's important, right? So these changes to your chromosomes, right? Not the actual A's and C's and T's and G's, right? We're not changing the words, but the chromosomes themselves and how they wrap around those proteins are changed. So imagine if it's wrapped around a protein really tightly, it's going to be hard for the machinery to clamp on that DNA and read the code. We're going to talk about that later too. But if it's really loose, then it's going to be easier for the machinery to clamp on your DNA and read the code, right? So by changing other parts of the chromosome, not just the letters, you change how often these proteins, these genes are expressed, right? And the environment can play a role in these changes. So all of this stuff may explain how one identical twin can suffer from a genetically based disease where the other one doesn't. And again, I'll go back to my metaphor. Think of it as a book. If they're identical twins, they have the same exact books, right? With the same exact words in each one of the books. But if one of the twins has the pages stuck together where the instructions on how to, for that disease are, then that twin's fine. Because yeah, they have that disease, they have the gene for that disease. For whatever reason, that gene is not being expressed with the first identical twin. But with the other identical twin, because of the environment, their chromosomes are open in that spot. So we can express that gene. Hope that makes sense. Again, same exact genome, same exact genes. It's just that because of the chromosomes, because of the environment, the chromosomes have been modified to where some genes are expressed more than others. And once again, big picture here. We're talking about changes in the genetics, but without changing the A's, the T's, the C's, and the G's, right? So remember, those are your codes. Those are the letters of the genetic alphabet. And we're not changing that in this situation. We're just changing how easily the code is read. Any questions so far? I've got a little bit more information on that side.
Is it sort of like if both my parents are smokers, would there be a good chance that I'd be wanting to be connected with nicotine as a child?
I don't know. I do think I've read somewhere that smoking is one of those things, and don't quote me on this, you can look this up in Binword. I thought I read somewhere that smoking not only could mutate your DNA and change the code, but it also, I think, if I remember correctly, can also affect your epigenetics. I do think that smoking can change, chemically modify or remove chemical groups on the DNA, to answer your question. That's all I know about it. And again, it's been a while since I read that, so I might be misremembering it, even look that up for you. Yeah. But, which is a good segue into this, epigenetic modifications, and of course, changes in the actual gene activity that result from the change in the epigenetic modifications may be carried on to the next generation, like you just asked and I just answered. Now, is it specifically smoking do that? Nicotine, I don't know for sure. But in general, like if you ask a less specific question and a more broad question, then there's the answer. Sometimes things you do, can modify your DNA, not the A's, the T's, the C's, and the G's, but the other things we just talked about. And sometimes those modifications can be passed on to the next generation. But here's the good news. Unlike mutations, which are alterations to the DNA sequence, which is changing the A's, the T's, the C's, and the G's, unlike that, these epigenetic changes can be reversed. And again, we're we're learning so much about it, even though when I said recently on the PowerPoint, and I said it's not recent, I guess it still is relatively recent because, all the other stuff I was teaching you about, complete dominance, incomplete dominance, co-dominance, we've known about this for hundreds of years, right? Well, almost hundreds of years. And this is all relatively new information. So we're still learning about how things change your epigenetics and how it's inherited. I've even heard or read, maybe even something about mental stuff, like memories. I don't know, that might be complete BS. To me, it sounds like complete BS. But, you get bombarded with information, and unless I look it up and verify it, then it just stays in the back of my brain and it's like, hey, I read that somewhere. But that doesn't mean it's true. Anyway, any questions about epigenetics? Let me back one up. I circled a word on that screen, and that is the next word for attendance. It's on the right-hand side. I should probably not say I circled. I surrounded it with a red line. That's not technically a circle. I wonder if any math people ever get irritated about that. Like, that's not a circle. That's not a line. Sort of like I do in my family. It must piss them off all the time. Like, I've got a theory. I'm like, no, you have a hypothesis. Say, shut up. Anyway, now we are down to the last bullet point of this chapter, and we can actually probably go through this pretty quickly. Because your old textbook, not your old textbook, the old textbook, after explaining everything that I just explained with these first 2 bullet points, then the old textbook went back and revisited everything that they mentioned earlier, but then they set it in the context of the chromosomes, not just the genes. But we've already been doing that. I've been doing that as I've been doing these first 2 bullet points. So for that reason, a lot of what I'm going to tell you is going to be slightly a review. So here we go. Chromosome theory of inheritance states that genes are located at specific positions on chromosomes. Part 1. You already know that part, right? There was a slide that I showed you last week where there was 2 chromosomes. They kind of showed them horizontally like this. There was a pair of homologous chromosomes, remember? And they were like, gene A is right here, gene B is right here, gene C is right here, and it pointed out that that's the loci, right? Those are the locations. And it gave an example, like maybe this one was homozygous dominant, maybe this one was heterozygous, maybe this one was homozygous recessive. I don't remember exactly what the example was, but we did talk about this. You do know that genes are located at specific positions on chromosomes. And I keep using the same stupid example, which is completely fake as far as I know. I keep saying something like maybe the gene for back hairiness is at the very tip of chromosome number 17, right? And if it was like that on my genome, it would be like that on her genome, his genome, and every other human who has regular chromosomes, right? Because that's how homologous chromosomes work. For the whole entire human species, specific genes are located at specific positions on specific chromosomes. And unless something goes wrong, which it does, they're all the same for all humans. And again, just to remind you, step back from this for a second, remind you we do have different alleles, right? So we might have the same genes in the same locations, but we do have different versions of them, which makes us different from each other. Anyway, you also know that the behavior of the chromosomes during meiosis and fertilization accounts for inheritance patterns. Meaning, think about independent assortment, right? When your chromosomes, when your homologous chromosomes, those 23 pairs line up in the middle of the cell in meiosis 1, it's all random. They line up independently of each other. It's not like all mom chromosomes on one side and all dad chromosomes on the other. It's all independent. Remember, there's like over 8 million different varieties of how that can happen. And it's all random. Therefore, once they segregate during meiosis, the chromosomes that this gamete gets versus that gamete gets, it's completely random. And that's the stuff that accounts for the inheritance patterns. So finally, right here, it is the chromosomes that undergo segregation and independent assortment, like I just said, and like I've been saying throughout this chapter. And it is the chromosomes that account for Mendele's laws. So once again, we can go through this pretty quickly because unlike the old textbook, I've been saying this throughout the whole chapter. So again, this should be a review for you. But here's a visualization of what we're talking about. So again, using the same example that we used before when we talked about this dihybrid cross, where the 2 characters we were looking at were P color, whether it be green or orange, green or yellow, and also P shape. So whether it be smooth or wrinkly. And we kept mentioning how that's independent assortment because those are two different genes on two different chromosomes. So just because you get your mom, the female's allele for P color doesn't mean you got the female's allele for P shape, right? It could, but doesn't necessarily mean that. So this is everything we just showed you, except now they just drew little chromosomes to show you how that works. That's all. So if I were studying, if I were you and I was studying, I wouldn't even worry about this picture, but there it is. So once again, independent assortment says they don't always line up with all female chromosomes on one side and all male chromosomes on the other, like they have here. Sometimes it's, they're swapped. And once again, this organism, this is only looking at two homologous chromosomes, right? Two sets of chromosomes. So imagine this, but with 23 of them. Think about all the different ways. I actually did the math yesterday because I wanted to see, I think it would be really cool if there was a website where you go to the website and it's just constantly showing them switching and showing all the different variations. And I did the math. If there was a website that did that and it changed once a second, right? It showed you all the different variations. Once it would take over 3 months before you could see all the different ways that your chromosomes can line up with mom on one side and dad on the other. So we could, because there's more than 8 million ways that can happen. But again, if you're studying, you can look at this if you want, but all this is doing, once again, is just showing you that, it's the chromosomes that are responsible for all the things we were talking about earlier. It's the chromosomes that result in this 9 to 3 to 3 to 1 ratio if we're looking at a dihybrid cross. It's the chromosomes that lead to the 3 to 1 ratio if we're looking at a monohybrid cross. So on and so forth. So it's all the chromosomes. That's all we're saying. Any questions on that? All right, here's where it gets a little bit trickier. And this is stuff I haven't taught you yet. Linked genes. Let's talk about this. Linked genes are genes that are located near each other on the same chromosome. And because they're on the same chromosome, they tend to travel together in mitosis. So for example, if gene 1 was for, and again, I'm just making this up, this is not true. If the gene 1 is for hair color, I put H for hair color. Say your mom, and this is the chromosome from your mom. Let's say she's got red hair. And let's say actually, you know what? I'm going to do that one. Gene 2 is for H. We'll call it that one for hair color. Gene 2 is for hair color. And let's say Gene 3 is for freckles, right? Whether you have freckles or not. And again, I'm just making this up. And let's say your mom has freckles. Now, if that was your mom's gene, chromosome, I'm going to try to draw your dad's chromosome here. This is meiosis 1. They're right next to each other. He's got gene 1 in the same location. He's got gene 2 in the same location. He's got gene 3 in the same location. He's got gene 4 in the same location. But for him, let's say your dad, and again, the hair gene is there for him and the freckle gene is there for him. Let's say your dad has, black hair and no freckles. If that were the case, if those genes, and again, I'm completely making this up, this is not how it works, and for these two particular genes, I don't know where those genes are in your genome. Anyway, if that did work that way, then you could presume that if you got your mom's red hair, you'd probably get her freckles because you got her chromosome, right? Those things segregated, and you either get this chromosome or that chromosome. So if you get this chromosome, generally speaking, and I'll tell you the exceptions here in a second, generally speaking, if you get this chromosome, you're getting all those genes, right? All those mom versions of the genes. Meanwhile, if you get your dad's black hair, you're probably also going to get his lack of freckles. Because in this hypothetical situation, those genes are right next to each other on the same chromosome. So you're either going to get your mom's chromosome or your dad's chromosome. Does that make sense? You're either getting this one or that one. Therefore, they're linked. They're close to each other. The problem is that where it makes it more difficult, though, is that sometimes we have crossing over. Remember crossing over? So in all actuality, you usually don't get just your dad's chromosome or just your mom's chromosome. Very often you'll swap chunks. So you'll get a mom chromosome with a chunk of dad or a dad chromosome with a chunk of mom. In which case, yes, you might actually maybe you could get your mom's red hair, but your dad's lack of freckles, if there was crossing over right there on the chromosomes. But most likely, since those two were so close together, that would probably be very rare. Not only are they on the same chromosome, but they're very close to each other. Meanwhile, the gene 1 was for, I don't know, breath. And your redheaded freckled mom has bad breath, but your dad has good breath. And again, this is completely fake. It doesn't work this way. In this case, it would be even more likely, like even more, not likely, but more possible, much more possible for you to get your mom's bad breath, but your dad's dark hair and freckles, because those are a lot farther apart. So again, crossing over happens, but these two are really close together. So crossing over would have to be pretty close, or pretty, in that small area for you to get a variation. But here, those two are very far apart. Basically, for the exam, if you have anything where I show like some kind of Punnett square, and it's not 9 to 3 to 3 to 1, if it's some kind of weird other ratio, then just say, oh yeah, those genes are probably linked. So that's all we're saying. In a sense, linked genes is almost a variation on independent assortment, right? So yes, genes are assorted independent of each other. Like I just said previously, it's because they're on chromosomes. So it's the chromosomes that are actually sorted independent of each other. But this is kind of like taking a step back and say, okay, but keep in mind, there are some genes, obviously, that are on the same chromosomes, and they travel together. And then you step back one more time and say, but there's also crossing over, which makes it even more complicated. Anyway, any questions about linked genes? All right. Let's talk about sex determination in humans. This one's going to be really easy. And of course, there are exceptions to this too. And also keep in mind, we're talking about sex, not gender. If you want to look that up for independent work and talk about the distinction between the two, you can. But in many animals, including mammals, we have a pair of sex chromosomes, as you already know, that are designated X and Y, at least for humans. You already know this. This is a review, but here it is again. Males have one X chromosome and one Y chromosome. Females have two X chromosomes. Yeah, that's it. I already told you that. I told you needed to know it, and there it is again. And as you do know from other stuff, there are exceptions, right? Sometimes a female might have 3 X chromosomes. Sometimes a male might have an extra X chromosome. Sometimes a male might have an extra Y chromosome. Sometimes a female will just have an X chromosome and not even have the second chromosome. It'll just be an X and a dot. There are variations, but this is the basic definition of sex determination in humans. And of course, like I said, if you want to look this up for independent work, there are exceptions to that. Just like with epigenetics and things like that. So you might have the genes, that might make you more male than female, but maybe those genes aren't turned on as much because of epigenetics. You might have the genes that make you more female than male, and maybe those genes are constantly turned on. So you're extremely more female than male. What do I mean by that? I mean, whatever characters you consider male versus female, like testosterone versus estrogen, things like that. Any questions about this slide? Pretty simple. I've already told you this before. Here's a picture of what we're talking about. There's the X chromosome on the right, the little tiny Y chromosome on the left. That's what they look like. So speaking of sex chromosomes, let's finally talk about sex-linked genes. And after that, I'll probably download the next PowerPoint and at least get that started. Sex-linked genes. These are a gene that's located on a sex chromosome, right? So every other genes that we were talking about that are on your autosomes, which are, chromosomes number 1 through 22, those are all just regular old genes. Sex-linked genes are simply genes that you find on your sex chromosomes. More of them are found on the X chromosome than found on the Y chromosome. Anybody not overthink this, take a look at the screen and tell me why we have more genes found on the X chromosome than the Y chromosome.
X is a little bigger.
It's bigger, yeah. So using the same metaphor that I've been using all day today, you can think of your chromosomes as a book. Each one of your chromosomes is a book that has a set of instructions on how to build proteins. This book is a much bigger book than this book, right? So that book's probably going to have a lot more pages, a lot more words on it, a lot more genes. Yeah, and that's it. Sex-linked genes are genes that are found on your sex chromosomes. But I can make it a little bit more complicated than that. Some human conditions, including red-green color blindness, hemophilia, and a type of muscular dystrophy, result from sex-linked recessive alleles. I'll put it next to this bullet point. Only indicating there's not going to be any questions where I expect you to know this. I don't need you to memorize sex-linked genes, right? I'm not going to ask you list the sex-linked genes, sex-linked recessive alleles. I'm not going to ask you this. These are just examples. You just need to know that yes, there are some of them that are on your sex chromosomes as opposed to regular ones. But nonetheless, we'll talk about it. And I know we have at least one colorblind person in here, so they can probably relate to this. Red-green color blindness is it's a common sex-linked disorder that is caused by a malfunction of the light-sensitive cells in the eyes. Again, this is all an example. You can study it if you want, but I'm not going to ask you any questions about it. Usually it's males that are affected. And also heterozygous females do have some defects. And the next slide I'm going to show you shows you the test. And again, once again, just to remind you, this is just an example. You do not need to know this. But before we move forward, based on these 2 bullet points, right here, see if anyone can figure this one out. And I guess you do need to overthink this a little bit. Based on those 2 facts right there, do you think that this allele, this gene, is found on the X chromosome or the Y chromosome? It's usually the males that are affected, and heterozygous females also could have some defects. They're obviously definitely carriers, but they can also have some defects.
Wow.
Good guess. But no, but why did you guess why? Because that would have been my first guess too. Exactly. And I just said mostly males are affected, right? So here's the key to how the giveaway here is that second bullet point. I guess I was a little bit tricky in that by pointing at both of them. Heterozygous females, right? So for a female to be heterozygous, that means she's got 2 copies of it, right? Therefore, it must be on the X chromosome. Because females have 2 X chromosomes. And also, if you back it up a little bit and think about it that way, the reason males are mostly affected is because males only have 1 X chromosome, right? So if they have it on that X chromosome, they have it. There is no heterozygous for them. They either have it or they don't. Anyway, can anybody, this is a little bit harder because of the lighting. Can anybody not read what number is on that screen right now? Just out of curiosity. Yep. Anybody else? So you guys can all read that number 7? Right there. Number 7. Yeah. And that's a good test for red. It's even more pronounced on the screen here. But anyway, because they're located, whoops, wrong way. Because they're located on sex chromosomes, sex-linked genes and exhibit unusual inheritance patterns. Again, like we just talked about with the whole male versus female thing with this one example. Since this is on the X chromosome, and males only have 1 X chromosome, then they don't need 2 copies of recessive allele. Because if they had that 1 copy on that X chromosome, that means 100% of their alleles are recessive, right? So they might not have 2 of them, but nonetheless, they have 100% of their alleles are recessive because it's on the, in this case, because it's on the X chromosome. I'll give you some examples here. Again, using the same example. Here we have a completely normal female, right? She's got her 2 X chromosomes. That's how I know it's a female. She's homozygous dominant, right? So she's completely normal. She's not affected. She's not a carrier. Here we have a colorblind male. You know, the X chromosome has that recessive allele. And once again, this is why I think last time we talked, I wanted to move away from saying to have the recessive phenotype or condition, you have to have 2 recessive alleles. Remember, I changed that last time we talked, and I transitioned into saying to have the recessive phenotype or the recessive condition, then 100% of your alleles need to be recessive, which used to sound like the same thing, right? 2 of your alleles are recessive versus 100% of your alleles are recessive. That sounded like the same thing, but now you can see it's not the same thing, right? Because here, if it's on the X chromosome, a male only has 1 copy. So all a male needs is that 1 copy, and they'll have the condition. Because 100% of their alleles, even though they only have 1, is recessive. Anyway, you see the Punnett square here. You know, when this female produces her eggs, those things segregate. But nonetheless, all she has to offer is an X chromosome that has the dominant allele. When this male goes through meiosis, those things segregate. So 50% of his chromosomes are X with that recessive allele. 50% of them are Y. Then you look at all the variations, right? So here we have this, there's 1 in 4 chance of them having a daughter who is a carrier, right? Because she has 1 dominant allele and 1 recessive. There's a 1 in 4 chance that they will have a male who can see normally. Oh, another 1 in 4 chance that they'll have a female who's a carrier. So 1 in 4, 1 in 4, that's half, right? 50% chance that they'll have a female who's a carrier. And then yet another 1 in 4 chance that they'll have a male who is not affected, right? So again, nothing to memorize here. But if I were to ask you to do a Punnett square, this is how you would do it. And you should have practice with this. Or you did get practice with this in the lab. But again, you can see here, even if a colorblind male, even though he's completely colorblind, he has children with a completely normal female, other than mutations, there's a 0% chance that any of their kids will have the color blindness. And of course, their females, their daughters, will be affected, and they will be a carrier, and they won't necessarily have color blindness. Here's just another scenario. We'll go through this really quickly. Here we have a normal male, unlike last time we had a colorblind male, and this time he's mating with a carrier female. Not a completely normal female, not a colorblind female, but a carrier female. Once again, when she goes through meiosis and produces eggs, those things segregate. So 50% of her eggs are going to have the dominant allele, 50% of them are going to have the recessive allele. When this male goes through meiosis and those things segregate, 50% of his sperm are going to be an X with a normal, with a dominant allele. And of course, 50% of them are going to be Y chromosome. You bring those together, you can see here, there's a 1 in 4 chance that they'll have a female who's completely normal and not a carrier. You can see there's a 1 in 4 chance that they'll have a male who's completely normal. There's a 1 in 4 chance that they'll have a female who's a carrier, and a 1 in 4 chance that there's a male who's colorblind. And again, there's nothing to study here, nothing to memorize. You do not need to memorize this, but if I were to give you this information and said this person mated with this person, what are the chances of their kid being this? You need to be able to figure that out. So any questions about that example before I give you one last example? Nope, okay. Last example here. Now we have a carrier female who's having children with a colorblind male. Once again, she goes through meiosis. Those chromosomes segregate. So 50% of her eggs have the dominant allele. 50% of her eggs have the recessive allele. This male goes through meiosis. Those things segregate, right? So 50% of his sperm have that recessive allele on the X chromosome. 50% of his sperm are just the Y chromosome. Bring those together. There's a 1 in 4 chance that they will have a female who's a carrier, a 1 in 4 chance that they'll have a male who's not affected, a 1 in 4 chance that they will have a female who is colorblind, and a 1 in 4 chance that they'll have a male who is colorblind. One last time, I want to remind you, do not memorize this. You could if you wanted, I guess, but if you get a question like this, I'm just going to give you the pertinent information. Excuse me. Say this person mated with this person. What are the chances of them having a daughter who's colorblind? What are the chances of them having a daughter who's a carrier? What are the chances of them having a colorblind male? What are the chances of them having a normal male? Or if I wanted to get real tricky, I could say, what are the chances, with this example, what are the chances of them having a completely normal female? And the answer would be 0, right? Because of the 2 females that they would have, one of them is either going to be a carrier or one of them is going to be colorblind. So without taking into account mutations, there would be a 0% chance for that couple to have a completely normal female. Woo! Any questions about that? All right. I guess the next word for attendance will be carrier. Carrier is the next word for attendance. I underlined it. All right. Let's talk about this very quickly. because we've got about 3.5 minutes left. Hemophilia was the other example. Once again, before I talk about this very quickly, I will remind you, I'm not gonna ask you any questions about hemophilia, but let's talk about it. It's a sex-linked recessive trait that has a very long, well-documented history. Basically, hemophiliacs are what we call free bleeders. You can read that and see why. What's interesting to me about this, and the only reason I even keep it in my PowerPoint, because as far as this semester is concerned, it's very unimportant, is that it's changed history. I don't know how much you guys know about European history, but they had the revolution in Russia, and that would not have happened. Well, there's a lot of reasons that it happened, but they had sick children with this hemophilia, so they brought this guy named Rasputin in, who was supposed to take care of their sick children. The guy was really weird. The people of Russia were like, what is up with this weird dude living with the royal family? And because that, amongst other things, led to them revolting against the family. Anyway, that also brings up another good point. If you're into history, you can look that up for independent work. What are some examples where times where biology has changed the course of history? It's happened a lot. I mean, there's a whole book on it called, I think it's Guns, Germs, and Steel, which talks about how the Europeans had conquered so much of the world. A lot of that had to do with diseases and things like that. Anyway, I left this part into, this is from the old textbook. You don't have to read about it, but you can if you want. Once again, I know people find dogs a little bit interesting. This kind of gives a background between dogs. Again, read it if you want, but it's unimportant as far as the exam is concerned. At the very end of this PowerPoint, you can see basically a review. So we talk about, well, yeah, in this case, it's just showing you meiosis, showing you fertilization. Yeah. So I guess the last word for attendance, even though I just gave you one, is going to be Callie, like the name Callie. It's in honor of my niece who's having surgery at 10 a.m. this morning. Nothing deadly. Anyway, that's it. I will see you if you want. I will be here and online at 10 a.m. for lab. But since you guys have so much lab stuff to do, you don't have to come in if you don't want to. Get caught up with your last lab report. But I'm here if you need me.
How do you spell Callie?
CALLIT.